Well, welcome everyone to today's by invitation webinar, which is hosted by the Center of Regulatory Excellence, or CORE, at the Duke-NUS Medical School in Singapore. We're delighted to have participants joining us virtually in Singapore and also around the world from CORE's advisory board, visiting experts, and adjuncts, and our various partner organizations and stakeholders, overseas regulatory agencies, the Singapore Ministry of Health, Healthcare Institutions and Health Sciences Authority, Duke-NUS colleagues, PhD students, and of course, our CORE graduate certificate students. I trust you are all keeping safe and well in these extraordinary times. So with the COVID-19 pandemic spreading rapidly, capacities of healthcare systems around the world are being massively challenged. But at the same time, global scientific research into COVID-19 therapeutics has been moving forward at an unprecedented rate. A wide range of test kits are being produced, new or existing therapeutic candidates tested, and at latest count, there are more than 70 teams working on developing a vaccine against the SARS-CoV-2 virus. To ensure that the world can benefit from all these developments, key scientific and regulatory issues need to be addressed to ensure that populations and patients can have access to safe, high quality, and effective products. Singapore has been actively involved in these initiatives, and so we're delighted to have our 6 eminent speakers today to share their perspectives. So with that, without any further ado, I would like to hand the time over to Dr. Sydney Yee, who will be talking about diagnostics and innovations locally and globally. So over to you, Sydney.
Thank you, John. And thank you to CORE for inviting me to this webinar. I just want to give a very general overview of some of the factors and the technologies and the innovations that are involved in dealing with, I suppose, any sort of situation, and more so in the situation of this pandemic. And I suppose, as John mentioned, when we're talking about dealing with this pandemic, it really involves many different facets and aspects in terms of formulating a strategy. So strategy in terms of understanding the pathogen first and foremost, and then coming into detection, so that we are able to better isolate, better trace, and better treat. And of course, ultimately, we're looking at how to prevent. DXD Hub is a national platform of Singapore dealing with developing different solutions, focusing on diagnostics. So I'm just going to focus a little bit more on the part of detection. So in terms of detection, we are really looking at many factors. It's a function of many things, what sort of detection platforms, technology platforms we're looking at. The technology platform ultimately is going to be dependent on the type of use cases, the clinical utility of whether this is something that we want to use at the bedside and at the clinic, or it is something that is lab-based, more for high throughput screening and high throughput screening purposes, or is it going to be actual diagnostics purposes? So that clinical utility workflow, including nowadays, we are aware that even if you have a solution, everybody is dealing with the same limitation of supply chain, procurement issues, logistics issues. Even if you have everything, the shipment becomes an issue. So these are the, so in terms of looking at building a detection product, we look at the different factors involving every single aspect of that. So not just the technology itself, not just the clinical utility, but also look way ahead into how do we procure some of the supply chain, how do we solve some of the potential supply chain issues down the line. So where technology platform is concerned, of course, we're looking at technology platforms that are detecting either nucleic acid, antigen, or antibodies. And that determines, or rather that is a function of what sort of utility we're looking at, because depending on which phase of the infection, it may or may not be relevant to look at a certain type of analytes that we're detecting. I think some of the speakers later will be talking about the different phases, and you'll be seeing different analytes. The first and foremost, when we're really looking at detecting pathogen in a way that is diagnostics and accurate, the gold standard really is Rt-Pcr. We are also seeing technologies that are a variation of that. So we have isothermal amplification, which allows that direct pathogen, the nucleic acid detection to be done a lot quicker. We are, of course, also seeing a lot of sample to result type, very low, minimal hands-on time type solutions. We also are seeing a lot of direct DNA hybridization type method. Some of the innovations that we're dealing with are looking at directly hybridizing the DNA, but without amplification of the nucleic acid. Instead, you use an enzyme to amplify the detection. So it allows the sample to be less clean, so to speak, because when you're looking at anything that has to do with amplification of nucleic acid, you need the input sample to be very clean. So that brings us to the limitation that we're seeing globally now in terms of RNA extraction. Every time we talk about any type of detection of nucleic acid, it almost always involves clean samples involving extracted RNA. So when we talk about the whole workflow of involving supply chain procurement issues, just having an Rt-Pcr test is not sufficient. You also need to think about how do we solve the limitation in the supply chain issue in RNA extraction. So we're seeing a lot of bundling effort in terms of that. How do we bundle RNA extraction solutions together with an Rt-Pcr solution? We are also looking at innovations that are doing sort of RUO type RNA extraction reagents to bundle with some of the Rt-Pcr kits that the labs are already using. So the first product that we developed in Singapore was one of these nucleic acid detection tool based on Rt-Pcr. Although the platform is not new, it's standard. Of course, in this case, the pathogen was new, and we have the opportunity to work with various research institutes in Singapore and the clinical community. So that was really crucial, which allowed us to very quickly, even though the pathogen was very much unknown in the early phase of this pandemic, as soon as the sequence came out, it allowed us to very quickly develop the test. In a very, very timely manner, we developed that within a month. Of course, that would not have been done, would not have been possible without, I have to say that we do have a regulatory agency that is very knowledgeable and very receptive. So I think that collaboration between the many different agencies, the research institutes, allows an entity like ours, which specializes in putting the many different components together to very quickly build a product. So it allows us to best enable our capability to make the product get to market very quickly. So today, of course, the product 42 is now in most of the hospitals in Singapore and also in a dozen other countries around the world. So we've been fortunate enough to have very good collaborations around Singapore to be able to make that happen. So since then, we also have been involved in developing solutions based on some of the other innovations that I talked about earlier. Maybe I'll leave it at that, and in case there's any other Q&A later, we can address those specifically. Thank you, John.
Thank you very much, Sydney. It was a very insightful overview of what's going on in test kit developments, including what's happening in Singapore. I'd now like to pass the time on to Professor Jenny Lau, who will be giving us an overview of therapeutics against COVID-19. Over to you, Jenny.
Yeah, so this is a snapshot of all the current drugs that are in the pipeline for COVID-19 treatment, and as you can see, there are more than 100 of them. So I'm not going to be able to cover everything, but I will highlight some of the ones that are either very near the clinical or are currently in different stages of clinical trials. Next slide, please. So I very broadly classified the various drugs into 4 big categories. The first group are the investigational candidates. So these are the drugs that have not been registered or licensed for any other indications. So Remdesivir, we are very familiar with this drug because it's been heavily covered in the media. There are now multiple clinical trials that are ongoing in various stages of recruitment. This drug was originally tested against Ebola and marburg infection, but subsequently also found to have in vitro activity against SARS, MERS, and now SARS-CoV-2. It is a prodrug of adenosine analog, and it works to stop viral replication. The NEGM recently has a report on the use of Remdesivir on a compassionate basis and reported the results from the 53 patients that received Remdesivir. 18% mortality was reported, and to me that doesn't seem to be very much difference from the mortality rate that was reported in the early days from China. So we will await the results from the clinical trials. EIDD-201 is a novel compound developed by the Emory Group, and in vitro as well as mouse data showed efficacy, and according to the website, they plan to start the clinical trial very soon. If not, they might have started recruitment. Now, the rest of the 4 compounds listed, Mevrilumab, CD24FC, Lenzurumab, as well as Leronlimab, they are currently tested for various other clinical indications, including for graft versus host, cytokine release syndrome, and other inflammatory disorder, as well as metastatic triple negative metastatic breast cancer. The whole idea behind using all these compounds to treat COVID-19 is that the investigators believe these compounds can modulate the immune response during the cytokine storm that happens in severe COVID-19 infection. So I'll move on to the next category, convalescent plasma or antibodies. So on the use of convalescent plasma, the Chinese recently released a very small case series report of 5 patients by Shen et al. And they reported good outcomes in these 5 patients who were critically ill and went on to receive convalescent plasma from recovered COVID-19 patients. With good clinical outcome, fever, shorter fever duration, as well as reduce all the inflammatory markers. So having said that, it is not randomized. This is a small case series report. But there are other groups and pharmaceuticals that are looking to produce polyclonal hyperimmunoglobulin. So TAG-888 by Takeda. Takeda is partnering several other companies to manufacture polyclonal HIG, and again, if everything goes well, may enter clinical trials by late spring. And many other groups are working on discovery of antibodies from recovered COVID-19 patients, and I will not go through all of them. There are, Regeneron has a chimeric antibody derived from mouse, which target the spike protein. And again, based on the website, clinical trial is expected to start sometime this year. Locally, Tycan, a company that has been set up with funding from Temasek, has a fully human engineered IgG1 antibody also against the spike protein, and the work is moving on quite fast, and we may be able to bring the product to first in human as early as end of next month. So that's all I'll cover for convalescent plasma and antibodies. So we'll move on to the next group, the therapeutics that's been approved for other indications and now being tested against COVID-19 infection. So hydroxychloroquine was really in the limelight for the last 1-2 months. So originally an anti-malaria drug, and a check on the registered clinical trials registry, you can find at least 142 clinical trials testing this drug right now, but actually majority of them are not blinded. There are only about a third of the trials that are registered are proper blinded clinical trials. So this drug has, there is in vitro activity for SARS-CoV-2, and hydroxychloroquine we know has immunomodulatory effect on T-cell activation. It is used in chronic inflammatory disorders like rheumatoid arthritis. So it's thought that the use of these drugs may be able to suppress the cytokine storm through suppression of T-cell activation. Having said that, so far there have been 5 reports to date on the outcome of people who have gone on to receive hydroxychloroquine. And I'm not going to go through all the reports, but in summary, I don't find any evidence that this drug has any clinical benefit or has any effect in terms of viral shedding duration. Lopinavir, ritonavir, Kaletra is the trade name, and the open label randomized clinical trial result has already been published in NEJM recently. And again, well, the study number recruited was quite small, N was 199. I think the primary endpoint wasn't very well stated, but again, the conclusion from that study did not show any clinical benefit or any benefit in viral clearance. So in most places, as I understand in Singapore as well, we have stopped using Kaletra as a potential treatment for our moderate and severe COVID-19 patients. The next compound, tocilizumab and sarilumab, they are both IL-6 receptor and antagonists. Tocilizumab has been, there's a small case series published by the Chinese, N is equal to 21, and they showed that, again, it's not randomized, and is a case series report on the use of this drug in the critically ill patient. But in the case series, they show improvement in both clinical as well as the cytokine markers, including lung imaging, all showing improvement. So I think we need a proper clinical trial to look at the efficacy of tocilizumab. In the US, sarilumab is being tried as well, and I think there are phase 2 and 3 trials ongoing with a plan to recruit up to 400 subjects. So we will await those results as well. Last but not least of the therapeutics, I will just briefly touch on Ivermectin, because just very recently, the group from Monash University released their in vitro data on Ivermectin against SARS-CoV-2, showing remarkable inhibition just after 1 dose. Ivermectin is an antiparasitic drug, but it has been shown previously in vitro to also inhibit many other viruses, including influenza, HIV, and dengue. But we have not seen that translated into any clinical benefit. The trial that was tested against dengue stopped at phase 2, and I don't think the drug was subsequently tested in any large phase 3 trial for dengue indication. So we will await more data from the Monash group. Last but not least, I put there cell-based therapy. There are other groups also trying mesenchymal stem cell therapy, bone marrow stem cell therapy, as well as natural killer cell therapy. I will just leave it as that, and I won't go into the details of these cell-based therapy. So that will be all from me, John. Thank you.
Thank you very much, Jenny. I'll pass the time over to Professor Wee Eng Yong to talk about vaccine development. Over to you, Eng Yong.
Thanks very much, John. I'll go through these slides very quickly, and so that we'll have time for discussion. So this figure, this slide basically summarizes the different vaccine technologies that we have in play today. If you go from the left to the right in an anti-clockwise direction, the first three that you see on the left-hand side, live attenuated, inactivated, and split inactivated, essentially are not possible for coronavirus 2 because this is a BSL-3 virus. So if you want to produce any of these vaccines, we'll need a manufacturing facility with a BSL-3 containment. That'll be way too expensive. So what we're left over is everything else from synthetic peptides onwards on the right-hand side, and if you go in that same anti-clockwise direction. And each of these have got its challenges in terms of its chemistry, manufacturing, and CMC properties and all that. And for Singapore, we've chosen to go with the RNA vaccine, and if we have time, we can always come back and discuss the merits of that decision. But suffice to say that I think what part of it is that this has a chance of rapid manufacturing because the entire thing can be made without cells. Next slide, please. And really what we want to try and do is, this is fundamentally the challenge that we have, right? So this chart basically shows you from a point where you identify an antigen to the point where you get into first in human trials. Usually it's about 5 years, if not possibly even more. And then you go through, and that's only a safety trial. And then after that, you go through a series of other trials to show immunogenicity, to show efficacy and all that before licensing. And the entire journey takes 10 years, if not more. And you're trying to shrink that down, the entire 10-year process, to something like a year. It's a major challenge, right? And so besides the, finding the vaccine and taking it through non-clinical tox studies into humans, on this little inset that you see in purple, you see the number of people that we usually need or normally need to test vaccines. So starting with phase 1, which is somewhere between 20 to 100, to phase 3, often in the thousands. Just to give you a ballpark, for dengue, Sanofi needed 30,000 people to be enrolled into a clinical trial to determine the efficacy. So that's costly, and it takes time to enroll that number of people. Next slide, please. And so how we're going to come at this is essentially to try and make use of tools that give us an earlier answer. So what you see in the top left-hand corner, the challenge right next to it, the outbreak, clinical trials in months, that's the Ebola outbreak in West Africa in 2014. The curves that you see in red, green, and purple are in the 3 countries that had these outbreaks. And the bars that you see in gray are the clinical trials. And you can see that the trial started when the outbreak is past its peak and on its way down. And so essentially many of these trials had no answer. And part of the reason is because of this on the right-hand side, the number of people that needed to be enrolled in the trial. But for everything that we know about vaccine in terms of immunogenicity, i.e., whether it produces the right immune response or that it produces side effects, everything has a molecular basis. And so what we've been trying to do is to understand what is that molecular basis that drives immune response, and the molecular basis behind common side effects like headaches, body ache, and fever that we sometimes develop transiently after vaccination. And if we can understand and if we can identify those molecular signatures, then these appear before the symptoms, before the immune response. And by tracking these, we will be able to, at least make some educated decisions on which vaccine to really invest our limited resource in. So that's an overview of vaccine development for COVID-19. Thanks, John.
Thank you very much, Eng Yong. Let me now move to the regulatory aspect of the webinar and start with Dr. Setharaman Rama. So Rama, over to you, please, to talk about supporting innovation and regulatory approvals for diagnostics.
Thanks, John. Good afternoon, everybody. Next slide, please. So talking about COVID-19, when we say regulators, people always relate to regulators as gatekeepers, but there's another important role we have been playing, which is in supporting timely access to safe innovations for our patients. That's one of the key role of regulators, which Sydney had also alluded to in our partnership with DXD. So we had embarked on this journey a few years ago for the medical devices side in HSA, setting up our pre-market consultation pathways where innovators could talk to us, discuss their regulatory compliance strategy with us. And we had partnered early with DXD under that for the last couple of years. So with all the experience we have had, when the COVID-19 came up on the agenda, we jumped in straight away. So what we had done is we had engaged with lots of these developers and discussed with them on the COVID-19 test they were developing. So we discussed both scientific and regulatory aspects and provide inputs for them to consider. So this includes could be like, how do you validate? How do you make sure that the test works as intended? What are the minimum studies you need to do? How do you manage false negatives? What do you think about in terms of your device design, in the test kit design itself, and various aspects? And it also includes what exactly is the intended purpose. Are you looking at diagnosis? Are you looking at detecting markers? And various such aspects. And we have had this kind of discussions with many such groups of developers, which has been very positive and enriching. Next slide, please. So this was our discussions and what we have been doing to support the developers. And then the next step, which is to pay timely access for these diagnostics. So we came up with what we define as a provisional authorization process. Is basically a fit-for-purpose regulatory pathway because there is an urgent need for diagnostics. So WHO says test, test, test. So we need to have those tests available. So in order to make sure that we can have reasonably safe and effective diagnostics on the ground for our population, so we sat down and we came up with what we call as a risk-calibrated review process. So we set up minimum essential requirements that we want to see before a test kit can go on to be used on the patients. And then based on that, so some things we did differently here is like we leverage alternate approaches. So for PCR tests that Sydney had spoken about, so we leverage a lot on in silico analysis of the design of the primers, probes, and the target sequences. And based on that to know that, yeah, okay, there's reasonable inclusivity. The test is able to detect majority of the viral sequences out there. We leverage also for cross-reactivity on top of lab testing, the wet bench testing, which is the most common way. Lots of blast results from databases. And then, of course, this is the part what we see before we allow or issue an authorization. But the most critical part is that we have a very sound post-approval monitoring requirements in place. So we are quite cognizant that we have looked at the minimum essential requirements. And in the current situation, the virus is mutating. There are thousands of sequences now published in the databases, and you are having new sequences being published for the virus, the mutations and information. So HSA also has post-approval requirements for these applicants to come back and report to HSA on a periodic basis on things like viral mutations and whether it impacts the assay performance, their respective assays. They are also required to report any inaccurate results they become aware of. And then they are also required to come and report to HSA. These are additional requirements on top of what we normally require for IVD tests. And we at HSA can also monitor some of these evolving scientific information and look at the bioinformatics data that's coming up and to see whether any of the tests that's being approved could also be impacted. So it's a more holistic approach, leveraging post-market requirements and then using them to support the pre-market provisional authorization process. The web link here has more information on our provisional authorization pathway. And the interesting fact is that when we started, the first few tests, what we approved under the PA pathway were all local companies or local developments, which was a very heartening sign to see. And we work closely with them to take them through this process. To date, we have over about 15 or so, or more than 15 tests that we have already approved under this pathway. Next slide, please. That was about the diagnostics aspect. Then coming over to the patient management or therapeutic medical devices, HSA also has looked quite early in the time point, and we have put in place certain regulatory flexibility to improve access to essential medical devices. So the first devices that we all are hearing about, the respiratory support devices for COVID patients. So what we have done is we have put up a three-pronged strategy to improve and facilitate access to these devices. One is we have many, based on global experience, although this has not really come live here, we noticed that a lot of manufacturers and hospitals are trying to use anesthesia machines and other positive airway pressure devices simply to augment the need for more breathing support for patients as a number of cases are increasing. So what we have already put up preemptively is that to allow where such devices, the manufacturers come with specific instructions and support to allow these to be used as breathing support or emergency ventilators. We are allowing safe use of such devices. We have also jumped in to say that, okay, if there are registered ventilators out there, these devices continuously undergo upgrades and changes. So what we have said is in this period, you don't have to wait for HSA's approval. As long as the the specifications of the ventilators are intact, it's working as intended, they can go ahead to implement the changes, upgrades, new accessories, so that as Sydney shared, sometimes it's not just about approving, but we also have to think about what is available at the time point when the whole globe is fighting for the limited resources that's available out there. So that's the second strategy. And the last but not the least, we are also cognizant there will be essential unregistered new models which should probably be available for supply for our patients. So we've also set up a separate pathway where companies can approach us and we will facilitate access to such devices. The link here also has further description of these initiatives. Yeah, I will leave it at here and then we can discuss further for other aspects of regulatory approaches. Thank you.
Thank you very much, Rama. Now over to Mr. Fu Yang Tong, who will be talking about the regulatory support for accelerated access and facilitating international product development and trials. Over to you, Yang Tong.
Right. Thank you, John, and thank you to the organizers for putting this call webinar together. Well, I mean, after hearing the speakers sharing their perspectives with regards to medical devices, with regards to therapeutics and vaccines technology. Yeah, and also looking at some of the questions that we have received, it's like, I think we may have a sense of being overwhelmed with the scale and the extent of information that is changing on a very rapidly and on a daily basis. Yeah, so I thought that this webinar is very useful to have everyone come together and really share what we know now and what we are doing and how could we sort of like collaborate and work together in combating this threat that the whole world is facing, perhaps except Antarctica. Now, what I'll do in the next 2 or 3 slides is just to share what HSA is doing with regard to advancing and safeguarding public health in the context of COVID-19. Yeah. So Rama has very nicely already mentioned that HSA is being, aside from being a regulatory gatekeeper, but it's also very importantly to that we advance public health and to make available medical devices, therapeutics products in a very timely manner, and especially more so in the context of COVID-19. So very first thing is to facilitate timely access to essential critical health products to combat COVID-19. But at the same time, we also got to be very cognizant of ensuring reasonable safeguards on the safety, quality, and efficacy of those health products. And very importantly also that we cannot be trying to do this alone. I mean, we heard from the speakers earlier that one key message that comes across very clearly is that the need to collaborate, not just within the local agencies and stakeholders across the researchers across companies that are developing devices or therapeutics, but also to have very close international cooperation and collaboration with regards to contribution and keeping abreast with the various scientific developments that are happening so fast. And the last point here, I thought I just want to share something that when we talk about the science and technology, and I must say the very pioneering innovation that has been happening that we have not seen for, I believe this is the first time at such unprecedented scale and speed of development of devices and therapeutics, that it's also very important that there must be an authoritative advice and information on health products to members of public and the healthcare professionals. Now, so in this context, I'll just briefly talk about this part here, that actually misinformation that is being shared to the public. And hence, in this context, HSA has issued advisory, which is on the HSA website, as well as on the MOH website, that that we advise consumers on products claiming to prevent or treat COVID-19. So I think this is also important to take note that it's not just that we are trying to, at one end, ensuring there is timely access of new innovative products that are for COVID-19, but at the other end, we also have to make sure as a public health agency that we advise consumers of misleading and of claims with regards to products that can treat COVID-19. Yeah, and on this note, I think we do active surveillance, and we issue warnings to suppliers and dealers who make such fraudulent claims. And we also work at the back end with website listings with the website administrators to remove inappropriate and non-compliant postings. And on this, and also we handle a lot, quite a significant number of queries. Initially, I mean, almost close to more than 400 queries in just the first 2 weeks of February. Now, next slide, please. Yeah, so with regard to facilitating clinical trials, we have expedited 3 clinical trials on remdesivir as listed here. Yeah, but I just want to say something about the second one, which is, I mean, the first one, Jenny has shared quite a fair bit. I just want to say something about the second one, which is sponsored by the US NIAID, and it's being conducted here. And this is a study that we should promote because with regards to this is the adaptive design, where the study will actually compare different investigational therapeutic agents to a control arm. So in this context, if one therapy proves to be effective, then this treatment becomes the control arm for comparison with newer therapeutic agents that comes subsequently. And on this note, so if there's any change to the design, then or to the drugs that are used, the sample size will also be updated. And aside from these 3 trials that are listed here, there are potentially 3 more investigational therapeutics coming online, I hope quite soon. Yeah, I think Jenny has alluded to one of them earlier in her presentation. Yeah, so I think at HSA, the Innovation Office and the Clinical Trials Branch has been meeting with researchers and developers for these other products that should come on board quite soon. And these are first in human studies. Yeah. I just want to add that this is not normal times. It's not business as usual with regards to an application process for clinical trials. So on this note, I mean, at the third point there, we adopt a rolling submission strategy with regards to trying to expedite review of COVID-19 trials. So this is one of the questions that a couple of participants have asked, that how is HSA managing the accelerated review to COVID-19 trials. So in this regard, I think we open. We allow companies to provide us their data set, their plans if they don't have the data as ready yet, so as to give us an overview of what's their plan like. And for some companies, they are also, not just with regards to the non-clinical studies and the clinical trial design for the first-in-human studies, but some companies are also establishing manufacturing facilities in Singapore to develop or to manufacture the products here. So in this regard, we also advise companies on the manufacturing controls that is required to ensure that the product manufacture is of appropriate quality to support that first-in-human study. Yeah, I mean, I'm sure we all heard in the media that the level of unprecedented scale and speed of development. At the same time, I would also like to put things in perspective and in context that speed has to be seen with regards to control of quality that I think Sydney and Rama has also alluded to in the context of medical device with regards to the importance of having to validate the devices for use in clinical in patients. Yeah, that the devices are reliable. And similarly for therapeutics, that we will need to have control with regards to the quality and the safety. And in the context of vaccines, I think we've got to be very mindful that we are using the vaccines in healthy volunteers, so that benefit risk assessment has to be carefully considered. Yeah, but nevertheless, in this context, it's important that we work across the various government partners, the ministries, the agencies, and to be able to rapidly move forward to develop therapeutics and devices to treat COVID-19 patients. Next slide, please. Yeah, so this is my last slide. So I just want to say something about the need to have not just a local collaboration, but also having that ability to connect with other regulators. That this is not Singapore's problem. In fact, this is a serious threat to global health. Yeah, that we work across with other regulators. In fact, we have a couple of meetings with the various regulators, as some of which are mentioned on this slide. In very early days, in early February and even end of January, we have contacts with the agencies mentioned that, US FDA, EMA, Health Canada, with regards to exchanging information for COVID-19. And this is the to facilitate the development in Singapore, and as also to do it in a very coordinated manner across the various jurisdictions. And on this note, we are also participating in the various platforms. For example, one of my colleagues is a co-chair of the Vaccines Expert Group at WHO. The other co-chair is from the US FDA. We are also a member of the COVID Therapeutics work group that is convened by NCID. And on this note, I think Jenny has given us a very nice overview of the global development of therapeutics that is being currently considered. And but I just also want to add on a bit more with regards to in a local front. What are we doing? Because I think one of the questions, in fact, a couple of questions are asking the same thing, that there are many products available with potential benefit, but at this juncture, I would just like to iterate that currently there are no drugs approved for COVID-19. Yeah, and there is a global regulators' consensus that we all need to, that no medicines has yet to clearly demonstrate efficacy in treating COVID-19. And hence, we would like to stress that the fastest way to serve patients is to collect robust evidence to determine whether either investigational agents that is coming out for first-in-human studies or repurposed medicines, of which Jenny has mentioned quite a couple, that will be safe and effective for treatment of COVID-19. and the best way to do this is to do multi-center randomized studies to generate the evidence to enable rapid development and approval of potential treatments for COVID-19. So this is an approach that the regulators have agreed upon, and we also like to share with our stakeholders and developers for therapeutics and vaccines. Yeah. So in this regard, we have in fact a couple of meetings at the international front that it's under the umbrella of the ICMRA or ICMRA, which is the International Coalition of Medicines Regulatory Agency. There were 3 workshops, virtual workshops, that have been conducted from March to just 2 weeks ago. So 3 workshops. The first workshop was for vaccine development, which is a very important area that to tackle COVID-19. And as we see from the news, that there are more than 70 over candidates that are being developed for COVID-19. So, and hence it's very important that regulators, developers alike have to come to some form of agreement as to what's the best way to move forward, and to be very coordinated and organized, and have to, with regards to at least the non-clinical requirements that is needed to bring forth their candidates for a first-in-human study. Yeah, so I mean, and hence this was an approach that we agreed. And as you can see, I think there are already now 4 or 5 vaccines that are already in the first-in-human studies. So in the Singapore's context, we hope to see one very soon, as I think you saw in today's paper. Yeah, also we are working very closely with the researcher on this. Yeah, and also we had a second workshop that talks about treatment, which I think I won't go into much detail because Jenny has covered quite a fair bit. But suffice to say that this is a threat that is affecting all countries, and hence that the regulators express the need to collect robust and reliable evidence. And the best way is through a randomized control trial with an appropriate control arm. Yeah. So I'll end on this note. Yeah, so thanks very much.
Thanks very much, Yang Tong. And now to round off the presentations, can I invite Dr. Tippy Mak to talk about balancing the benefits and risks as we prepare for the rollout of COVID-19 vaccines. Tippy, over to you.
Good afternoon. Thanks, John, and thanks to the core team for the invite. I have a happy talk because I'm going to make some happy assumptions that we have successful clinical trials. We can fast forward to 2021, and many thanks to Eng Ong, Jenny, and Sydney, we have good vaccines and tests, and we have expedited reviews from Yang Tong and Setharaman. And I'm going to assume that Singapore has a healthy supply of COVID-19 vaccines. So my job is to give you a future sense of what a mass COVID-19 vaccine program would look like in Singapore. And I want to emphasize why we must prepare now. And please consider that we must keep thinking about the end-to-end vaccine development, what the target program, what the target vaccine will look like. And so I'm ready on my first slide. The key questions we should be asking is, what are the characteristics of the first generation COVID-19 vaccine that we hope to see? Now, this is taken from the, sorry, my slide's a bit different from what I had, but it's on the bottom, you'll see the WHO website, which shows the target product profile, TPP, which I find extremely useful, and I'd suggest you look at it in full. It was developed in February by a group of experts, and it's continuing to be refined. I extracted the latest version, which is 9th of April, and I am giving here just the highlights of some of the characteristics. I emphasize it's not an official WHO guidance, but or document, but it's giving us some good guidance on about what we should be looking for. It gives a framework for all stakeholders, from the public, the researchers, policymakers, regulatory and pharma colleagues who are listening in, how we can frame what is a benefit-risk profile, because as you know, every vaccine that will emerge successful from the clinical trials will have its own unique benefits-risk profile. It sets reasonable minimal expectations, and all the noise and all the discussions and details, I think we can continue to refer back to this TPP. Just want to add, it's not the only TPP that WHO has provided. It also has provided one for Ebola, Nipah, MERS, and Lassa. Any accelerated vaccine program, there's a TPP. If you look at the first column, you see the vaccine characteristics. In the second column, here is the ideal preferred characteristics that we would have, where there's no pressure from the outbreak. In the third and final column, you see the critical or the minimal characteristics that we want to see as we are now in the midst of an outbreak. I want to first look at the middle column. Please stay with me at the preferred characteristics. The ideal vaccine actually protects against the virus, SARS-CoV-2. You don't get the disease. There are no contraindications, maybe rare allergies for some, but that's for all vaccines. And it's indicated for all ages, including children, because we want to actually have adequate population herd immunity to prevent epidemics. So it would be ideally a single dose. And you can see this is a very similar profile to the seasonal influenza vaccine program that we currently have. Please, the next slide. If you stay in the middle column for the preferred ideal vaccine, what are we aiming for? We want at least 70% efficacy. And this is with consistent immune protection with the elderly who are at higher risk. And of course, we want rapid protection within the usual 2 weeks. The duration will be at least a year. And if you look at the safety and reactogenicity profile, a highly favorable benefit risk with no serious adverse events and no enhanced disease. If we can go back to my previous slide, please. Okay, now let's look at where we are, the minimal vaccine characteristics in an outbreak setting. So the TPP shows us that we want in our first generation COVID-19 vaccines at least protection against the disease, against COVID-19. It may not protect us against the virus actually getting infected, but we want to see protection endpoints on severe disease, hospitalization, ICU deaths. We want to reduce those. You may get the disease, but it should be a milder form. In an outbreak vaccine, some populations will not be studied. As Engel has pointed out, it's going to be thousands of recipients, volunteers. However, we are not going to be able to capture all subpopulations, and they will have to be studied after, perhaps. So there might be some contraindications. The target population I'd like to mention is focusing on the high-risk groups. And as we know, they're mainly adults. The higher risk with increasing age and existing comorbidities. We do need to vaccinate children because we want to have, ideally, good herd immunity. I also want to note, it doesn't mention here on the April 9th TPP, but if you looked at the Chinese data and the American data from the CDC, there is another high-risk group, and you looked at the children, the pediatric population, the children under the age of 1 were at higher risk for hospitalizations and severe disease. So actually, another high-risk group we should look at is this group, and we must also consider the safety of vaccination in pregnancy to confer passive immunity through placental transfer and through breastfeeding. So you see here also, hope you can see it, there's probably going to be a 2-dose vaccine. And the reason is with the new vaccines, as Eng showed the whole spectrum of different types of technology, these are highly purified subunit products. They're not that immunogenic, and most of the new vaccines have not been a single dose. Can we have the next slide, please? Okay, so you see now it's very flexible. If we even have a point estimate, even with the confidence interval between that might not be, ranging from 30 to 70%, it is still considered a vaccine that would be very useful because a 50% efficacy against the disease would actually benefit a lot of people and reduce all the hospitalizations. So this is an important factor that we're looking at a partially effective vaccine that's acceptable. If you look at safety, and the duration of protection could be less, we look at safety for the first generation vaccines in the context of an outbreak. Okay, if you look at, it says here, benefit risk assessment should take potential enhanced disease into account. This means that we need to quantify that risk. Now, ideally, this is one of the questions about antibody-dependent enhancement and what are the post-marketing commitments for new vaccines like this. We need to really be able to see whether we can find any sign of disease enhancement in vaccinees, even in the phase 2 and 3 trials. It really depends on the force of infection, how much the virus is circulating in the clinical trial sites, because we actually could get quite empowered data if it's in a high transmission site. So this would be very valuable to know whether we are going to see any sign. We must answer that question: is there enhanced disease possible with any of the vaccine candidates? Now, I can think of 2 examples very briefly. With RSV vaccines that were done in the 1960s on infants, we saw very early on in just a few dozen babies that there was evidence of enhanced disease. That is, the vaccinated cohort were more likely to be hospitalized than the non-vaccinees. If, for example, mentioned with CDV, TDV dengue vaccine, we needed long-term, large population studies to be able to find that. So we need to actually answer this question carefully, quantify it, use mathematical modeling to help understand the benefit-risk profile. Yes, we can change the slide, please. Okay, that was 2021 in the future. I'm now going to, if you're a Star Trek fan, put you back into the transporter, and we're going to go back to 2009 because I find this is very important. Many of you listening, including myself, we have some experience from the past pandemic where there were new vaccines. And I want us to remember all the things that we did that time and the key considerations. Okay, we need secure global manufacturing capacity. I want to remind that in 2009, more than half of the world's H1N1 vaccines were in the hands of a few countries. We had inadequate supply for the world. Indeed, some countries I shouldn't name even forced their local manufacturers to renege on commitments, international commitments, to ensure first their own domestic supply. So we need to do this better this time. The stakes are much higher. This is really a pandemic on fire. We also need to have a good global access policy. You know, Seth Berkley from Gavi, Bill Gates, many are calling for this, a clear global level policy about equity. And I think this is something that needs to be inked at the G20 level. And again, we did not have this in 2009, and we can really improve from that. We are going to be facing new technology, and we did also in 2009. If you recall, we had the oil and water adjuvants, MF59, ASO3, and these, we had cell culture-based vaccines, we had live attenuated intranasal vaccines. This was a challenge to convey messaging to the population, to prepare them, and to trust in the science of these new technologies. With COVID-19 vaccines, as Ellen has shown with all the possible vaccines, there are a lot more technologies that we need to be able to convey to the public in a clear manner. We need standard research protocols, case definitions of adverse events of special interest, adverse events of following immunization, both predicted and unpredicted. Good examples I put in the sidebar in the bottom. Guillain-Barré syndrome, we knew from the 1976 pandemic that there was an increased risk of Guillain-Barré and neurological acute demyelinating disease from the vaccine. There was an association, a clear association. So when 2009, this was a clear adverse event of special interest, and we had to follow closely and look at background rates of expected Guillain-Barré over observed in the vaccinees. And this, again, is here we now have ADE as a concern, as an adverse event of special interest. Also, we have adverse events following immunization concerns, and we had unexpected ones in 2009. That was the ASO3 adjuvanted flu vaccine and narcolepsy that was first discovered and described in Finland in a cluster of children who received that vaccine. So this was completely unexpected, and we should be prepared for the unexpected. Finally, I want to emphasize that we need to leverage technology better. We need real-time data, both in the real-world effectiveness and the safety monitoring. So to answer that question, what are the long-term commitments? We have long-term commitments with these new accelerated vaccines, and we should be able to, every Singaporean resident should be able to easily register any concerns about the vaccine. We should be easily able to link vaccinees to any hospitalizations. And I think we can really leverage where we are now with smart technology, and Singapore really could lead in this area. Next slide, please. Okay, this is my final slide. I wanted to give the key takeaways in a nice Doris Cohn orange color. that to stay grounded, please use the framework of the WHO target product profile. I think it's a very useful framework to help have these discussions. We need to prepare now for rational recommendations and prioritizing the special needs groups, the high priority groups. We need to have adequate program planning now and use master protocols, which I mean the international protocols, standard case definitions to look for adverse events following immunization. And I want to highlight that we really need a very effective communication strategy. We can draw on a lot of lessons we already learned from the 2009 pandemic. I think Singapore has been a leader in many ways, and it's a model for pandemic management, interministerial task force, which has shown us, how it's a whole of government approach. We need the structures because in a vaccination program, we will definitely be looking at workplaces and schools for vaccination. This could be the biggest world vaccination program we've ever seen. So I think Singapore's potential to be a global leader beyond its own borders is very high. We can see lots of innovations, technology used to monitor this vaccination program in unprecedented ways and have really good long-term follow-up for effectiveness and safety monitoring. That's all for me. Thanks. Happy to take questions.
Thanks very much, Tippy. So in view of the time, what we're going to do is that, in fact, there have been questions coming through the Q&A function. And in fact, along the way, panelists have been answering. And we will leave the Zoom function on until about 2:30 so that you can go through the answers. There are some questions for which there are no answers yet, and we will try and link up the right people to answer. But if you're anonymous and you don't want to be identified in the Zoom function, you can email us separately and we can get the answer sent to you. What we've also asked the panelists is because things are evolving so fast. In fact, they are prepared to come back at a later date as things are developing. We may have a part 2 to this webinar to give you updates on what's going on. So that may in fact be a better way to deal with the time that we have. But what I'd quickly like to do is to just round up today's webinar, which has been very rich, is to ask each of the speakers in turn, give us one takeaway that you would want us to remember from today and in the context of everything that's evolving now. So Sydney first.
Yeah, just very, very quickly. So we do see and evaluate a lot of many different innovations. We all try to do our level best to deal with this pandemic. Just want to maybe remind the innovators out there that when we think about an innovation, do think from the perspective of the whole solution, especially in this time when the supply chain really is the main issue in a lot of these innovations.
Thank you. Jenny?
Hey, John. So I think trying to run clinical trials in a pandemic environment is extremely challenging. As we can see, like I said, most of the trials are not conducted in the gold standard manner of a double-blind, placebo-controlled trial design. So it makes the interpretation of the results extremely challenging for us. So I absolutely agree with Yang Tong that, while we're trying to balance speed, because that's what we urgently need, we have to be very mindful as well in terms of how we conduct the trial so that data can be interpretable at the end of the day.
Okay, thank you. Eng Yong?
I'd say, be grateful for your support in many ways. As Jenny said, when we're getting into phase 1, phase 3 trials, if they're volunteers, it'll make the journey a lot smoother. And of course, funding support, right? These things need funding, and if you can help in either way, and also the advocacy just to get the word out to the public when we're ready for the trials, all those things will help very much. Thanks.
Thank you. Rama?
Sorry. So if you have innovations, if you have solutions that you have done reasonable work on and you're confident that you've done your part, I think I would invite all of you you can approach HSA. We will be more than happy to work with you to facilitate access where it is going to be useful and safe for our patients. Thank you.
Thank you. Yang Tong?
Right, thanks, John. Yeah, I just want to also add that come to us early, even you don't have data yet if there are plans to conduct studies. Give us an overview, your plans. We will definitely meet up and arrange a meeting as soon as possible. In fact, we have been meeting researchers and stakeholders very frequently in the past 1 month, as well as going forward, there'll be more of such meetings. So one thing I think I also just want to emphasize that is the, well, I think we need to focus on the science and ultimately the data, the data that will be informing of us in order to provide evidence, and so that we as a regulatory agency, whether it's for medical devices or for therapeutics and vaccines, is that we are able to make informed regulatory decisions. And the thing also, I just want to add that while how fast, or rather, I think some of the questions that are coming in is how is CHSA expediting it? Yeah, in fact, we are not, as I mentioned in my slide presentation earlier on, it's not business as usual. We are handling and reviewing the applications at a very fast pace. And I must say it was done, I think, for those cases there within a very, very short time. So rest assured that it will not be the usual timelines that we have. Yeah, so I just want to emphasize that as well. Thank you, John.
Very good, Yang Tong. And Tiffany, your last word, please.
We need to work very closely and prepare early to the public to understand these new vaccine technologies, that to prepare them that we may only have partially effective vaccines, the way science works. And if we find in the end, with knock on wood, very good vaccine candidates that become good vaccines, we can still fail if we do not properly execute a good communication strategy and an effective vaccine program.
Okay, thank you very much. So thank you very much to all the panelists. As I said, if your questions have not been answered, because some of the earlier questions that you sent to us by email have already been addressed in the presentations, others have come through in the Q&A chat while the presentation has been going on, and we've sought to answer those. And we will seek to respond to those along the way. It's a very challenging time, but many, many opportunities, as you've heard. The innovation boundaries are being stretched. As you can see, not only the researchers, but the regulators are responding to this very proactively, not just in Singapore, but all around the world. And so for all of us who have a stake in the game, and actually all of us do, because COVID-19 affects us all as a pandemic around the world, I think we need to watch this space, play our part, and be very proactive, but also optimistic as we look to therapies, vaccines, and test kits. So on that note, thank you very much for joining us this afternoon. We will keep you informed of subsequent webinars and a potential follow-up to this one as well. And stay safe, keep well, and we look forward to seeing you soon. Thank you very much, and good afternoon.
Thank you.